Hello and welcome back to Survey of Biology. So this is Unit 10, Section 1, where we're going to start our discussion of genetics. And if we look at the history of genetics, one man stands out kind of as the father of modern genetics, an Austrian friar named Gregor Mendel, who worked with pea plants in the mid to late 1800s. And he had a great analytical mind and kind of the time and the resources to look at literally thousands, if not hundreds of thousands of pea plants, individual crosses, and look at the traits. So remember, this is a time where they didn't know about chromosomes, they didn't know about DNA, and his research really laid the groundwork of what we know now. And so why did he choose pea plants? Well, four big reasons. First, they had many traits that were either or, so he could really develop his ideas of dominant versus recessive traits, whether they would show up or not. The second was that he could control mating. He could literally take a small paintbrush and take the pollen from one pea plant and pollinate a second pea plant that he wanted to. So he could control the traits that he was working with very easily. The third one is that he could develop these true breeding lines. So one trait that we'll look at is flower color in pea plants, and purple was dominant over white. And he could create a line of plants that he knew always produced purple offspring. He also created a line of pea plants that always created white offspring for the flower color, so he could create true breeding. And then last, and maybe the most important, is he could get a large sample size in a relatively short amount of time. He could have 2 or 3 generations within a year, plant the seeds, grow them, and go through that process several times and literally get data that was just almost overwhelming in terms of the number and the ratios that he was able to develop. So as he was doing that, he had a kind of a vocabulary that's very different than what we might say in terms of terms for genetics nowadays. So he talked of things in terms of unit characters. So these were the heritable features like hair color, or with the pea plants that he was looking at, flower color or seed shape, seed color. And the second thing that he talked about were traits. So each variant for a character. So within flower color, we might have purple or white. Those would be traits of the unit character. And so in terms of our own vocabulary, we all know about genes and we talk about genes, but he didn't know anything of them. And so in terms of Mendel's vocabulary, Mendel's unit would have been what we call a gene. And here's a small table that shows some of those traits that Mendel had found in pea plants that were either dominant or recessive. So stem length. The pea plants were either tall or they were dwarf. There wasn't an in between. The flower position was either axial at the end of the stems, or axial in the middle, or terminal at the end of the stems. And seed shape was either round or wrinkled. One that's not shown up here that we'll talk a lot about is flower color. Purple was dominant over white. And so what he was able to do is develop his terms where dominant was a trait that was always expressed when present. And then recessive could be masked by the dominant trait. Even if a pea plant possessed the recessive trait, it didn't always show up in their physical appearance. So here's those true breeding lines that I was talking about. So true breeding pea plants produce offspring identical to the parents. So he was able to develop a true breeding line of purple flowered pea plants. He also developed a true breeding line of white flowered pea plants. And what he found is when he crossed that true breeding purple with the true breeding white is that all of the offspring in this F1 or first filial generation were purple. And from that, he was able to deduce that purple is dominant over white. So he had his first idea that these different traits, genes, one was dominant over the other and could mask the expression of one. So in terms of humans, if we look at some dominant and recessive traits that we might be familiar with in terms of our face, some of the ones that we might have already looked at a little bit is brown eyes are dominant over blue eyes. Freckles are dominant over no freckles. A widow's peak, some of you may have hair that comes to a point on your forehead, is dominant over no widow's peak. And we'll do a lab in class where we'll look at many of these human facial features. Now those traits, as Mendel called them, are what we call nowadays alleles. And for any trait, humans and mammals get a maternal chromosome from mom and a paternal chromosome from dad. And on those chromosomes, they code for the same traits. So for pea plants, this might be for flower color, but we have one allele, in this case for purple from mom, and in this case white from dad. So those alleles can be different. So what is an allele? Well, alleles are different forms of the same gene. Alleles of the same gene usually seem to be identical and differ just at one or few nucleotides. There are two copies of alleles of the same gene in diploids, one at the maternal chromosome locus and another at the paternal. So you have one from mom, one from dad in the diploid cells. So remember we talked about haploid versus diploid when we talked about mitosis and meiosis. So a simple example to remember, gene coding for the lip shape has two forms of alleles. Let's say an allele big A codes for broad lip shape and an allele little a codes for thin lips. So alleles are different versions of the same trait. So here's an example of two chromosomes, and we'll call the top red one, we'll say is maternal, and the bottom one we'll say is the paternal one. And the first one here is a gene for P, maybe we'll call this purple flower color. And this one happens to have a dominant P from mom, a dominant P from dad. So this is what we call big P, big P. This is homozygous. So the prefix homo means the same. So it's homozygous for the dominant allele. So here is the second trait, A, and this one is recessive little a, and this is what we call homozygous for the recessive allele. So there's a different possibility. And at the third gene locus, the location right here, if we look at B, it has a dominant gene from the maternal side, a recessive gene from the paternal, and its type of genes are called big B, little b, and this is heterozygous. So this means a mix of the genes. So when you have one of each kind, that's called heterozygous. So there are two different alleles in a heterozygote. Okay? So let's look at that a little bit in terms of something they call the genotype, which is literally just the type of gene that you have. So the term homozygous, as we said, means you've got two of the same allele. So in this case, if we're using T's, that could be big T, or little t, it doesn't matter. Now, if it's big T, this is going to be homozygous dominant, two of the same. If it's homozygous recessive, then it's little t. And the mix, or the heterozygote, right here is one dominant and one recessive. So there's our big T, little t in that case. So what's the difference between them? Well, the difference is in the type of genes that they have. So a homozygous trait is made up of two of the same alleles. So it can be dominant, big T. It can be recessive, little t. And in this case, little t, that's the only way that we're going to produce the recessive effect. So in the case of humans, blue eyes is a recessive gene, and the only way someone can have blue eyes, if they're little b, they have to inherit two recessive genes for that. And a heterozygous trait is made up of two different alleles. So again, just kind of learning the vocab a little bit and being familiar with those. And in terms of talking about mixing in two of them, now let's talk about hybridization. So one thing Mendel did was he made hybrid plants. He took two different traits, in this case, purple flowers and white flowers, and made a hybrid. So hybridization is when mating or crossing of two varieties takes place. And so Mendel started off very simply with doing something called a monohybrid cross. So he was looking at one character. In this case, we're going to look at flower color. So if he took two flowers, one purple, that was true breeding, one white, that was true breeding, and crossed them, as we saw before in that F1 generation, he got all purple flowers. Okay? And then, so that showed that purple was dominant over white. But then the interesting thing happened, if he took two of these F1 hybrids and he crossed them, the offspring always came out in this 3-to-1 ratio. He would have 3/4 of the offspring be purple and 1/4 be white. So the recessive trait, the white flower, skipped a generation and showed up back. But that F1 cross, the F2 generation, always resulted in this 3-to-1 ratio of purple flowers to white flowers, or dominant traits to recessive traits. Okay? And from that, he was able to kind of develop his law of segregation. So his law of segregation states that the separation of alleles takes place into different gametes. So if you have an individual that was homozygous dominant for purple flowers, when they produce gametes, if you remember from meiosis, those have to split, and he can only pass on one of those alleles. And if he's homozygous dominant, the only thing he can pass on is big P. Same thing is true if it's little p, little p. The only gametes that they can pass on would be a little p. But if they are a heterozygote, he could pass on big P or little p, but not both. So the law of segregation says that he can only pass on one allele and that they'll separate. And the last law that we need to look at in terms of Mendel is something called the law of independent assortment. So here we're going to look at more than one trait. So we're going to look at a dihybrid or trihybrid, so 2 or 3 traits. So the genes assort independently. So in this case, the T for tall and Q for quasi-colored could be different genes. And they independently segregate of pairs. There's an independent segregation of pairs of alleles during gamete formation. So what that means is that if we've got these 4 genes, the T's will always be split up and the Q's will always be split up. So there's a possibility of getting 4 different types of gametes. We can have big T, big Q, big T, little q, big Q, little t, little q, but we'll never have the T's together, we'll never have the Q's together. So that's the law of independent assortment. So there we go with some basics of Mendelian genetics. We'll pick up in the next section when we talk about genotypes and phenotypes, and then get into Punnett squares and how it can apply to humans. So thanks again for your time.